Good evening everyone, and welcome to tonight's Research Tuesday event, Memory Restored. My name is Nick Drevis and I'm the Marketing Manager for Research and Innovation here at the University of Adelaide. Firstly, I'd like to acknowledge and pay our respects to the Kaurna people, the original custodians of the Adelaide Plains and the land on which the University of Adelaide's campuses at North Terrace, Waite and Roseworthy are built. Tonight we focus on a devastating subject. Memory impairment is devastating not only for those directly impacted, but for families, friends, as well as groups and businesses who are unable to contribute towards. Memory impairments are common across a wide range of mental illnesses and strongly contribute to poor functional outcomes in these illnesses, including a reduced quality of life and even homelessness. Adding to the bleak outlook, there are currently no effective treatments for memory impairment. However, research at the University of Adelaide could be showing a way forward. To understand more about this research, we'll be joined tonight by Dr. Nigel Rogasch. Nigel is the head of Brain Stimulation, Imaging and Cognition Research at the University of Adelaide and at SAMRI. Nigel graduated with a PhD from Monash University in 2014 and has worked at labs in Melbourne and Adelaide, as well as in Canada, Germany and Turkey. Nigel's research focuses on combining brain stimulation and neuroimaging to study the neural mechanisms underlying cognition, the pathophysiology of mental illness and the use of transcranial magnetic stimulation to probe and alter brain function. Before Nigel joins us on the stage, I'd like to acknowledge the questions that have already been pre-submitted, and I welcome you at home to submit questions via the Q&A section on Zoom or on Facebook Live. So now I'd like to invite Dr. Nigel Rogasch to the stage.
Thanks a lot, Nick, and thank you very much to the Research Tuesdays team. It's certainly great to be here. I must admit the lecture theatre is looking somewhat reminiscent of the 8 a.m. biology lectures I used to attend here as an undergraduate. It's looking quite empty, but I trust that you are all enjoying watching this from the comfort of your own homes. So today, what I'd like to speak to you about, as Nick alluded to, is a little bit about some of the research that we are currently undertaking here to try and understand more about memory, memory function, how it works in healthy people, and what happens in people with severe mental illnesses and how their memory function can be impacted by their illnesses. I'd also like to talk a little bit about some of the research that we're doing to try and address these quite devastating deficits that people have. So this is the brain. Now, the brain is quite remarkable. Although it only accounts for about 2% of the total volume of the body, it uses over 20% of the actual energy. So it's made up of between 80 to 100 billion tiny cells called neurons. Now, 80 billion is an enormous number. So to give you some kind of context, the Milky Way, the galaxy that we live in, contains roughly 200 billion stars. So that means that all of us are walking around with the equivalent of a small galaxy floating around inside of our heads. So to add to the complexity, those 80 billion neurons form over 100 trillion connections with each other. And it is those connections that give rise to us. So they allow us to experience the present. So by integrating all of the sensory information that comes in through our senses, the brain creates this live stream of consciousness that we all experience. It allows us to experience the past by storing those experiences as memories and allowing us to relive those. And it also allows us to experience the future by looking ahead, planning, and dreaming about what the future might hold. So the importance of the brain and the function of the brain is perhaps most greatly demonstrated by what happens when something goes wrong with the brain. And in particular, mental health disorders are an incredibly devastating and challenging form of brain disorders that some people experience. So mental health disorders are quite common. So around 10% of the population, the global population, is thought to have either experienced or is currently experiencing some form of mental health disorder. And that is particularly represented here in Australia, where that estimate is actually a little bit higher. And those are remarkably large numbers. A lot of these disorders are incredibly devastating and lead to long-term disability. And in fact, mental health disorders as a class make up over 22% of the global health burden. These are some figures from 1990, and I guess one of the most worrying things about these figures is they're not really changing at all. So over the last 20 years, between 1990 and 2010, that number of 22% hasn't really shifted at all. That's because a lot of these disorders are incredibly difficult to treat, and they really do lead to what can be, for some people, lifelong disability. So let's try and unpack that a little bit and look a little bit deeper into what these mental health disorders are and how they're actually diagnosed. So unlike a lot of other illnesses, mental health disorders are what are known as syndromes. So that means that most of them are actually defined as collections of symptoms. Now this differs quite substantially to other illnesses, which are often diagnosed based on some kind of a biological basis. So a good example at the moment is what's happening with the COVID-19 pandemic. So if someone presents with a cough and a fever and maybe a bit of a runny nose, they can go and have a test to see whether that illness is, or what is actually causing that illness, whether it be just a simple flu or a cold, or whether it's something a little bit more sinister, like something related to the COVID-19 virus. So the difference with mental health disorders is that all of the definitions and the diagnoses need to be made just on the external symptoms themselves. And this is decided by the book which is shown up here, known as the Diagnostic and Statistic Manual for Mental Disorders, or the DSM version 5. So let's take an example here in terms of depression. So depression is a really debilitating and devastating illness that causes people to suffer from depressed mood for long periods of time. So to get a diagnosis of depression, what you need to do is to meet 2 of these 8 different criteria, with depressed mood being one of those. So what you can see by reading through this list here on your screen is that there's quite a number of different possible combinations that people could present with to receive a diagnosis of depression. So for example, item 3 here is you could present with either significant weight loss or significant weight gain associated with increased appetite. So the challenge here is that this can create a mind-boggling number of different combinations of symptoms. So for example, in one study, a group of researchers surveyed 3,000 odd individuals who had depression, and they looked at their symptom profiles, so what different symptoms that they actually were showing at the time. And from these 3,000 individuals, this resulted in over 1,000 unique symptom profiles. And importantly, none of those symptom profiles were present in more than 2% of people. So this means that we have an incredible diversity of symptom profiles within these different mental health disorders. As I mentioned, another real challenge is then trying to link these symptoms down onto their biological processes. So what is incredibly challenging is often when we look at the brains of people who have mental health disorders, there's no clear and obvious pathology. So this is contrasted here up against another type of brain disorder. So this is an advanced form of dementia. So on the left-hand side, we have the brain of a healthy individual, and on the right-hand side, we have the brain of someone who has end-stage Alzheimer's disease. So you can see from this image that diseases like Alzheimer's suffer from a really large degradation of the brain and its tissue. You can see really quite advanced shrinking of a lot of the brain regions with this disorder. When we look at people with mental health disorders such as depression or schizophrenia, what we see is more typical, I guess, of what you would see of a healthy person's brain. Now, that's not to say that there aren't differences, but those differences tend to be a lot more subtle. So here is an example of a study done in people with schizophrenia. So schizophrenia is another very devastating mental illness that is characterised by psychosis. So these are a range of symptoms such as hallucinations, so seeing or hearing things that aren't there, and delusions and paranoia, and also thought disorder, so a difficulty in organising thought. So in this study, what the researchers did was they scanned using a magnetic resonance imaging machine a number of people with schizophrenia and a number of people who were considered healthy controls. So the authors compared the thickness of the brain between these two populations, and when they compared these at the group level, which is what you're seeing in the slide here at the moment, they were able to observe quite widespread reductions in the size of the brains in people with schizophrenia. So this could lead to the impression that schizophrenia itself is associated with widespread reductions in brain size. But what these authors then did is they used a specific statistical approach to look to see whether these changes in brain size were consistent across the schizophrenia population. And what they found was that they were actually remarkably inconsistent, to the point where most people showed a unique profile in terms of where their brain was actually thinning. And what is indicated in the bottom half of this slide is the areas that showed where a brain area that was smaller than would be expected from the healthy population in over 2% of those people with schizophrenia. So that shows that really not only are the symptom profiles quite diverse, but likely the underlying biological causes are most likely equally diverse. So this is leading to a rethink in the way that people are trying to classify and understand mental health disorders. So instead of going by the existing classification methods, which have served us well over the years, there is now a move to try and look more at the individual and the individual symptom profiles and the individual biology of those people to try and understand what is actually resulting in the symptoms that the individual themselves are displaying. So this is, I guess, a way of kind of breaking down the current classification systems to try and better understand how biology is mapping onto people's symptoms. So one set of symptoms that does appear to be quite common across different mental health disorders are what are called cognitive impairments. So cognition refers to a whole class of cognitive processes that are involved in us thinking. So for example, memory, attention, planning into the future, these kinds of activities. And when we compare people with, in this example, people who have schizophrenia and a group of people who have depression and bipolar disorder, what is clear is that there is quite a large reduction, particularly in memory within these people. So to orientate you to this graph, the horizontal line here indicates the level at which healthy people perform these different cognitive tasks at, and anything below this is a reduction in those symptom profiles. So what you can see on the left-hand side there, indicated by the red box, is the really quite strong and large reductions in these memory, the ability to remember different items. So what's really troubling about these cognitive disturbances and these cognitive deficits, and particularly the memory deficits, is that they tend to be very, very good indicators of functional outcomes from these illnesses. So they often are very closely associated with things like homelessness, unemployment, and possibly most worrying, quality of life as well. What's even more worrying is that currently most of the treatments that we have for these disorders don't have any positive impact on cognition. And in some cases, the medications themselves might actually make these cognitive impairments and these memory impairments worse. So to summarise, we don't have treatments for these memory impairments, and they likely contribute quite strongly to the functional deficits and the disability that these groups are experiencing. So this leads into some of the research questions that myself and my group and my colleagues have been asking over the last several years. So one of the key questions that we've been trying to answer is how does the brain actually store these short-term memories that are so deficient in these mental health disorders? So on the face of it, this may not seem like such an interesting question, but when we actually think about it a little bit more deeply, this is quite complex. So short-term memory refers to our abilities to take in information, hold that information in our mind for brief periods, perhaps manipulate that information in some way, and then retrieve the information and use it. Now, this is typically the kind of memory that occurs across very short time spans. So we're talking here in the sort of the seconds to minutes sort of time spans. So an example of this might be if you're walking down the street, your phone's run out of battery, so you can't access Google Maps. So you ask a passerby how to get to the Bragg Lecture Theatre. The member of the public might say to you, well, look, you just have to go left, you need to go right, and then you need to take another left. You then need to hold that information in your mind to actually walk down the street and execute those movements. And when we think about this a little bit more closely, this kind of ability actually underlies a lot of the functions that we undertake in day-to-day life. So even, for example, reading a book or reading a novel, so having to be able to maintain the words as we're reading a sentence in our mind to make sense of that sentence, of that paragraph, of that chapter, and then of the whole book. We need to be able to bring that information in and hold it. So in the lab, we tend to use, I guess, simplified versions of these memory tasks. So one example here is a task where we simply ask people to hold coloured squares in their memory for a short period of time. So what this allows us to do is to break down memory into some of its component pieces. So for example, when we are flashing the display up and providing the information, we can then take measurements of what's happening in the brain when that information is being encoded. We can then take that information away for a period of time, in this case for say a second or so, and then we can see what the brain is doing in that state. And then we can represent the information again and ask the participant to make a decision as to whether the original stimuli have changed or not, whether the image is the same. And then we can assess what's happening during the brain while they're trying to retrieve that information. So even though these tasks are fairly simplified versions of what we use in day-to-day life, they actually generally correlate very well with things like fluid intelligence, so our ability to deal with newly arising situations. So when we have people perform these tasks, we typically take some kind of a measurement of what the brain is actually doing across these periods. And one method that we use quite extensively in my group is a method called electroencephalography, or EEG. And this is a picture here of Carolyn Berryman, one of the postdocs in my group, and myself in the lab. So Carolyn is wearing one of these EEG devices here. So it looks a little bit like a swimming cap, but the main points of interest here are the small little circular discs that you see within that cap. So these are electrodes that record electrical activity that's actually coming from the brain. So neurons, when they are communicating with each other, send off small electrical signals. And these signals are actually, although they are small, they're large enough that if we place electrodes on the outer layer of the scalp, we can actually record those signals. So the recordings that we get look something like this. And there's actually a remarkable amount of information in what I guess looks kind of just like a squiggly line. So what we can pull out from these signals are what are called brain waves or brain oscillations. So these refer to these rhythmic periods of activity that neural populations undergo. So if you can imagine these 80 billion neurons are something like a group of concert. And speaking of memory loss, I've actually just completely suffered one right then. So imagine them as a band, right? So what we have is, the band might just be chatting before they start playing some music, but then as they come together, they synchronize all of their component pieces to make this coordinated activity. Now, these rhythms represent that same process occurring in the brain. So when large populations of neurons come together and start firing together, they give off these strong electrical synchronous signals, which we can record as these brain rhythms or brain waves on the cortical surface. So these rhythms come at different speeds. And the one that I'm showing at the top here is a slower rhythm called a theta rhythm. And importantly, these different rhythms seem to have different functional roles as well. So theta is thought to be important for the prioritization of information as it's coming into memory, and then coordinating different brain regions to respond to that information. A slightly faster rhythm is called the alpha rhythm. So this occurs at around about 10 or so cycles per second. And this is thought to represent a suppressive signal. So this tends to get larger in areas of the brain that are not involved in the task, and might represent a way that the brain actually actively suppresses regions that are not important in what you're trying to do. There are also these much faster rhythms, which are generally called gamma rhythms. Now, these rhythms are thought to represent a potential binding mechanism, where populations of neurons that represent different features of a representation. So, for example, a tiger, there might be some neurons that represent or are sensitive to stripes. There might be some that are sensitive to the colour orange. There might be some that are sensitive to the general shapes that make up the tiger. So gamma rhythms are thought to be able to bring all of those different neural populations together to be able to create what we then perceive as a tiger. So we've known for quite a long time that these different rhythms are all important in us being able to store these short-term memories. And when we look at these in mental health disorders, we actually tend to see at the group level, again, that many of these oscillations are different in those populations. But what we don't have a very good understanding of at the moment is how, at the individual level, differences in these different oscillations contribute to people's memory performance. So we're currently undertaking a large trial where we are recruiting a large population of healthy adults to come and complete memory tasks in our lab while we record these different brain oscillations. And what we are looking to understand is to whether one or all of these different mechanisms can contribute to a given individual's working memory performance. And already we've got some interesting preliminary findings from this study. We're currently recruiting at the moment, and this will continue to the end of the year. But what we found is something that was quite surprising that we weren't expecting. So embedded within these strong oscillation signals and these strong rhythms is actually this background noise. And for many years, people kind of ignored this noise as exactly that. So just a noisy signal that probably didn't really represent anything of interest. But what we've found is that actually the noise itself is also a really quite a good predictor, or is quite strongly associated, I should say, with people's memory performance. And interestingly, some very recent biological studies have suggested that this noise is actually quite sensitive to the excitation and inhibition that underlies these neural populations. So this is like the yin and the yang of the brain, and really for the brain to operate well, this excitation and inhibition need to be in quite good balance. If they're out of balance, it seems to impact this noise level. So what we're looking to do moving forward is to expand our data collection out into different populations who suffer from different memory deficits. So we've already started doing this in older individuals who also undergo memory impairments with natural aging. And we're starting to do this as well now in different mental health populations, so people with schizophrenia and people with depression. Something else that we're interested in from this data as well is to look at people's genes. And we're trying to get an understanding here of how people's genetic risk for these different disorders can potentially impact some of these biological processes as well. So the genetic profile of mental health disorders is quite complicated, but what we can do is we can calculate scores that look at all of the individual genes that potentially make up risk for one of these disorders, and calculate a score that can give us a general sense as to what the genetic risk for an individual is. And we're interested here to understand whether these genes that make up this risk factor for these mental health disorders also contribute to changes in these biological processes. Another question that we've been interested in is which brain regions actually generate these oscillations. So one of the limitations of our scalp recording methods is that being a long way from the brain, it's actually quite hard for us to tell from these electrode recordings where the signals are exactly coming from. So even though we're recording them over a certain brain region, it's possible that with the way that electricity spreads through the head and through the skull, that different brain regions could be contributing to that signal at a given electrode. So we've been interested in exploring new ways of coming up with methods where we can actually probe different brain regions non-invasively. And one way that we do that is to use principles of electromagnetic induction. So this is the same principle that your induction cooktoppers work through, and it's based on some fairly fundamental physics principles. So the method is called transcranial magnetic stimulation. And through this method, what we do is we use tightly wound copper coils, and we pass a really, really fast electric current through those coils. Now, when we do that, what happens is we actually generate a magnetic field which runs perpendicular to those electric wires. Now, importantly, those electric wires are placed inside of a plastic case, so the person doesn't actually experience the electric current at all. But this magnetic field can actually pass down through the skull and through the scalp, and then can induce another small electric current on anything underneath it which conducts electricity. Now, the brain is an excellent conductor of electricity, so if we place these coils on top of somebody's head and run our current through them, we can actually then create a small current in the outside layer of the brain which causes any neurons in the vicinity of that current to fire. Now, this is actually quite remarkable when you think about it. So we're actually able to, without going inside of a person's head, we're actually able as researchers and clinicians to interact with the brain. So this is one example here of myself giving transcranial magnetic stimulation to a colleague. So in this case, what I'm doing is I'm targeting the part of the brain that controls movement. So in this case, the cortex, and specifically I'm targeting the area of the brain that actually makes the hand move. So I'm just going to play this video here, and you'll hear the TMS coil discharge. It'll make a little clicking noise, and when it clicks, I need you to watch the participant's hand down the bottom there. And what you should hopefully see is a small hand movement that's associated with the stimulation. So what we're able to do here is to actually non-invasively activate the brain, the part of the brain that controls the hand, and generate an involuntary hand movement. So this gives us, I guess, a way of moving around and directly stimulating different brain regions, and then trying to understand how they respond to the stimulation. So we have spent a lot of time over the last few years trying to see whether we can use our EEG method to record how different brain regions respond when we stimulate them non-invasively. And EEG can give us a really rich lot of information about what happens after we stimulate a region, what happens to that region itself, but also where does that activity spread to other regions. So this is just an example of some of the activity that we can observe after we stimulate a region where the little coil is indicated there. So this has been slowed down a little bit from what actually happens, but what is obvious from this little video is that not only do you get activity occurring where you stimulate, but you also get quite a lot of the brain actually lighting up in response to the stimulation, suggesting that we're not only able to access the area that we're interested in, but also broader networks that area is connected to. So it's taken us a long time to develop this technique. It's actually quite technically challenging given how sensitive our EEG recording equipment is. When we put this really large electromagnetic field near it, it causes all sorts of troubles with the recording equipment. So we've had to develop different software approaches and different hardware approaches to allow us to undertake this kind of research. But what we're finding is that we're actually able to non-invasively trigger these oscillations or these rhythms from different brain regions. And as we move across different brain regions, these oscillations actually look quite different. So this gives us a new method now that we can go out and actually causally map the ability of different brain regions to generate these oscillations. And again, we plan to do this now across a range of different mental health disorders to try and understand the contribution of different brain regions and their ability to actually generate these oscillations. So moving on to the next big question I think that we're trying to answer is, instead of just, I guess, trying to understand how the brain works with this method, can we actually use our stimulation approach to actually improve memory? Now, how would we actually do this? So for many years, people thought that the brain was actually hardwired, that once you got to adulthood, that the brain didn't really undergo any changes at all, and that it was quite a solid structure, almost like a rock. But what we've found out over the last couple decades is that that is actually quite far from the truth. And the brain is actually not hardwired at all. It is actually quite plastic, and it is continually changing and reorganising in response to what's going on around it. So the brain is actually very malleable. It's more like, I guess, more like Play-Doh than it is a rock. And what we can do is we can actually use our stimulation method to try and harness that natural plasticity of the brain to try and reorganise areas or circuits that are involved in different brain functions, such as memory, and try and improve their functionality. So to do that, what we actually do is we apply rhythmic stimulation. So this is called repetitive transcranial magnetic stimulation, or repetitive TMS. So in one example here, what we do is we apply the stimulation in these rhythms that we see during memory. So in this case, we apply the stimulation at these fast gamma rhythms, and then repeat that stimulation at the slower theta rhythms. When we do this in animals, and so for example in a brain region known to be important in memory in rats called the hippocampus, we know that we can induce really quite long-lasting plastic changes in those regions. This is already, and this has been used, I guess, over the last 20 or so years in humans using this non-invasive brain stimulation method. And it's already had clinical success. So, for example, in depression, we know that if we apply this kind of stimulation to the frontal cortex, a region that's thought to be involved in the emotions of, in the dysregulation of emotions, that we can actually improve depression symptoms. So what we wanted to try and understand was whether we could apply this to regions of the brain that are involved in memory to see whether we could potentially improve memory performance. And in some work led by a colleague, Kate Hoy, that I was involved with, so what we did is we applied this stimulation again to the prefrontal cortex, and we measured people's memory performance both when we gave an active stimulation and then when we gave just a placebo stimulation. So something that sounded and felt a little bit like the real stimulation, but it didn't actually change anything. And we got quite excited because what we saw in this initial study was that we were actually able to boost people's memory performance. So here on the y-axis we have the ability on a certain memory task, the black line indicating performance after we'd stimulated the frontal cortex, and the dotted line indicating performance after we'd used a sham stimulation. So we thought we were really onto something here, and we as you do, we got quite excited, and we arranged a number of replication studies to try and see whether we could make this effect happen again. And I guess to our disappointment, what we found was that the size of the effect, as often happens, got subsequently smaller in all of our repeated replication attempts. And we got a little bit disheartened with this until we looked a little bit closer at the data and we looked at, I guess, the biological changes that we were able to induce in people. And we saw that even though I guess at the group level we weren't seeing strong changes, for some individuals we were seeing quite strong changes, and they tended to associate quite closely with the biological changes that we were able to induce. So this made us think, well, maybe the problem is not so much that the stimulation can't be beneficial. Maybe it's the problem that we're using this one-size-fits-all kind of stimulation. So we pretty much gave the stimulation the same way to everybody. So a PhD student of myself, Kate, and Paul Fitzgerald's called Seung-Wook Chung took this up. And what Seung did is he actually recorded these brain rhythms and brain waves while people were undertaking a memory tasks. And he looked at these slow theta oscillations and these fast gamma oscillations within these individuals and looked to see how those two rhythms were interacting with each other. And what Seung found is that when he looked across individuals is that the pattern was actually similar but a bit different between different people. So for example, what you can see from this plot here is that big red blob is not quite in the same plot on the graphs from two different participants on the left and right here. And so when you look at this across a group of participants, the exact rhythms that we're actually providing the stimulation at, and that's represented by the red square here, are not actually that close to where the individual participants' natural frequencies are. So those are indicated by the little triangles. So what Seung did is he then actually individualised the stimulation frequencies to the person's individual rhythms. And what he found was he was able to actually induce quite long-lasting and larger changes in brain activity during a memory task within those individuals. And most importantly, he was actually able to improve memory and give a memory boost to about 70% of those individuals. Now, these were just healthy young people, but what was quite interesting, and again a little bit unexpected, is Seung also just provided a mood rating to these people. And what he found was when he actually applied the individual stimulation, he was actually able to boost people's mood as well. So we're really, really interested in this individualised approach now, where we think that actually tailoring the stimulation to an individual's physiology might enable us to give larger boosts in memory performance and possibly even in things like mood as well. And the next stage for this research now is to take this into a clinical trial where we plan on applying this kind of stimulation method in people with depression. So just to quickly summarise, what I've hopefully shared with you today and what you can take away from this presentation is that the memory is impaired in quite a wide range of different mental health disorders. And this is really challenging because these memory impairments often have a large functional outcome, and we currently don't have any really good treatments for these memory impairments. So what is also important is that the brain functions that are likely underlying these memory deficits seem to be likely to differ between individuals. So one-size-fits-all treatment approaches probably won't work for trying to rectify these issues and to try and improve memory. So hopefully using personalised or individualised methods such as our brain stimulation technique might provide a new way in which we're actually able to make some inroads into these memory deficits across these wide range of mental health disorders. So I just want to finish up by just quickly thanking all of my team and my collaborators who have helped out on this research over the last few years. If anyone's interested in finding out a bit more about the kind of research that we're undertaking, this was just a small snapshot of some of the research that we're currently doing. Please feel free to get in contact with me either on email, on Twitter, or you can visit my university website profile there as well. Thank you very much.
Thanks, Nigel, for that great presentation, and it's certainly encouraging to see some great results and look forward to the clinical trials. We'll now be answering some questions, so thank you to those who have been sending in your questions. We'll take the first one. I guess, is keeping mentally and physically active a good safeguard against dementia?
Yeah, so that's an excellent question, and certainly, I mean, one area that's of great interest at the moment is I guess the healthy aging process, and as I'm sure a lot of people have experienced, as we get older, we do just naturally experience, I guess, a degradation of memory over time. And I guess, you know, in disorders where that's accelerated, so disorders like dementia and associated disorders like that, obviously if there's things that we can be doing before then to prevent that decline, that would be really helpful. So, I mean, the answer to this question is not easy. It's a very, very active area of research that I'm certainly not an expert in, but some of my colleagues are currently working on this quite closely. And it seems to be that there are a number of, I guess, lifestyle factors that can really help slow this process down in some people. So examples include, I think as the questioner noted, so physical activity is a good one, sleeping well, eating well, maintaining social interactions, continuing to learn new things. So all of these different lifestyle factors seem to generate something which is generally called cognitive reserve. So this is this capacity to maintain your cognitive abilities as you get older. So I guess at the moment there seems to be good evidence for most of those. What we don't currently know is, I guess, what are the most important things to do, and colleagues like Ash Smith at University of South Australia and Mitch Goldsworthy here at the University of Adelaide are currently about to undertake a project where they're actually trying to understand what is the best combination of those different activities that will provide the best protection, I guess, moving forward against that natural cognitive decline, and hopefully as well against some of those more insidious declines that happen with things like dementia.
Another question around that is, I guess, what percentage is genetics and inherited, as opposed to, I guess, what's sort of environmental, e.g. addiction?
Sure, and that's that's another really great question, and unfortunately another one that has a fairly complicated answer. And I think, yeah, the answer is we really don't know at the moment. And again, this is another really active area of research, and I think there's some really fascinating findings that are coming out in this area. So it used to be very much a nature versus nurture kind of debate, where I guess our genetic makeup was thought to be quite separate from the environmental factors that we encountered as we go through life. But I guess what we're starting to understand now is that those two things are much more closely related than what we previously thought. So the field of epigenetics, for example, is showing that the way that our genes are expressed, so the way that our DNA is read, can actually change in relation to into experiences and environmental factors. So the answer is that it's probably a really strong combination of the two of those things. So difficult to put a percentage number on it, but it's likely that, you know, our genetic makeup will predispose us to certain things, but then it's how the environment interacts with our genes over time that will, I guess, lead us down the path that we end up going.
Yeah. Next question is, could personalised brain stimulation be used to improve memory in those who have memory loss associated with ECT?
Another excellent question. So to just back up a little bit on that one, so ECT, or electroconvulsive therapy is another form of brain stimulation that is commonly used, particularly for depression, but also in some people with schizophrenia as well. So this form of stimulation is quite different to the form of stimulation that we use. So the aim of ECT is to actually induce a seizure in the person that is receiving it. Now that sounds quite extreme, but it is actually a really, really effective treatment for depression. And for people that where things like antidepressant medications don't work, and if they have really, really treatment-resistant symptoms, often this is the only kind of treatment that will work for them. So the downside to this form of treatment is that it's often associated with quite severe memory impairments most likely related to the seizure. So although it works really, really well for the depressive symptoms, it has this common side effect which people often find equally as disabling. So it's a real trade-off. And I think, certainly areas that we are becoming increasingly interested in is whether we can combine these different forms of treatment together to find, I guess, ways that give us the maximal impact of both while minimising the bad outcomes, I guess. So I think, potentially using our non-invasive form of brain stimulation with different kinds of treatments could be quite useful. Another one that we're interested in and looking at the moment is whether if we combine brain stimulation with some of the lifestyle factors, so for example with physical exercise. So if we combine those two things together, can we get better bang for our buck out of both of the I guess treatment options to really give people as big a boost as we possibly can.
Yeah. Now, I don't know if I'm going to pronounce this correctly, but do programs like Lumosity and Brain HQ mitigate memory loss?
Another really interesting question. So again, just to give a little background to that question, one way in which people have thought might be useful in trying to improve memory is to train your memory. So I guess playing games, and I think Lumosity is like an online platform where different games have been developed that are thought to I guess that require your memory. So it's thought that if you keep exercising your memory using these kind of training games, that might be one way in which you can boost your memory and maybe improve your memory and maybe sort of stave off some of these cognitive declines that happen with aging. So again, it's kind of a bit of a complicated answer to this question. It's sort of, I guess, probably 10 or so years ago, there were some really positive trials that came out that sort of suggested that these cognitive training programs might be really, really useful. Since then, there's been a lot more research, and as often happens, the picture looks a little bit less clear than what it did initially. So one finding that seems to be coming out fairly consistently now in some of the larger studies is that the gains that you get from that kind of training often only occur quite narrowly in the kind of cognitive domains that you're specifically training on, and sometimes even actually only the tasks themselves that you're training on. So if you train on say, for example, trying to memorize colored squares, you get better at memorizing colored squares, but then that doesn't help you, say, trying to memorize a phone number or something like that. So it's sort of, I would say, unclear at the moment as to whether those kind of those programs are beneficial, or whether they're maybe not being as beneficial as what we initially thought that they might have been.
Yeah. Excellent. Well, that brings us to a close for tonight's Research Tuesday event. I'd like to thank Dr. Nigel Rogasch for his informative presentation, and thank you to everyone at home for tuning into tonight's presentation. So take care and stay safe, and good night.